Welcome to Chapter 2, Drug Discovery-Specific MI Methods. In this chapter, we'll explore the technical foundations of AI-driven drug discovery, from molecular representations to generative models. You'll learn how molecules are converted into computer-readable formats and how machine learning is applied to predict drug properties. The first step in drug discovery, MI, is converting chemical structures into formats that computers can understand. This choice of molecular representation significantly affects model performance. We'll cover four main types: SMILES strings, molecular fingerprints, three-dimensional descriptors, and graph representations for neural networks. SMILES, which stands for Simplified Molecular Input Line Entry System, is the most widely used format for representing molecular structures as strings. Basic rules include atoms like C, N, O, S, bonds where single bonds are omitted, double bonds use equals sign, triple bonds use hash, ring structures marked with numbers, branches expressed with parentheses, and aromatic atoms in lowercase. Let's look at some SMILES examples. Ethanol is written as 3 characters, C, C, O, 2 carbon atoms followed by an oxygen, which the notation reads as a hydroxyl group. Aspirin shows an ester and carboxylic acid. Caffeine contains a purine scaffold with methyl groups. Ibuprofen includes an aromatic ring and chiral center. SMILES advantages include compactness and human readability. Disadvantages include no three-dimensional information and potential for invalid strings. Molecular fingerprints represent molecules as bit vectors sequences of zeros and ones. They're widely used for similarity searches and QSAR. ECFP, extended connectivity fingerprints, work by starting from each atom, hashing neighborhood structures at different radii, and converting to bit vectors. Different variants use different radii, with typical lengths of 1 to 4,000 bits. Different fingerprint types serve different purposes. ECFP with radius 2 is great for similarity searches and QSAR. Larger radius provides more precise structure recognition. MACCS keys use fixed substructures for fast searching. RD-Kit fingerprints use path-based approaches. The Tanimoto coefficient measures similarity between fingerprints, ranging from 0 for completely different to 1 for identical molecules. The Tanimoto coefficient is calculated as the intersection divided by the union of 2 bit vectors. Practical thresholds are: very high values mean same compound class, high values mean similar with possible similar activity, moderate values are somewhat similar, and low values mean different molecules. This is essential for virtual screening and compound clustering. Three-dimensional descriptors quantify the spatial structure of molecules. Major types include molecular surface area like TPSA or topological polar surface area, which predicts oral absorption. Volume and shape descriptors include molecular volume, sphericity, and aspect ratio. Charge distribution includes partial charges and dipole moments. Pharmacophore features identify hydrogen bond donor and acceptor positions. Lipinski's rule of 5 uses key descriptors to predict drug likeness. The criteria include thresholds for molecular weight, lipophilicity measured by log-P, hydrogen bond donors, and hydrogen bond acceptors. Compounds satisfying these rules tend to have high oral absorption. This remains one of the most important filters in drug discovery. Graph representation treats molecules as mathematical graphs where vertices are atoms and edges are bonds. Each atom vertex has features like atomic number, atom type, hybridization, formal charge, aromaticity, and hydrogen count. Bond edges have features like bond order single, double, triple, or aromatic, and whether they're in a ring. This representation is ideal for graph neural networks. Now let's discuss QSAR, quantitative structure-activity relationship. The basic hypothesis is that similar molecular structures show similar biological activity. This is the similar property principle. The general QSAR equation is activity equals some function of descriptors. Activity means a measured biological activity, such as IC50, or the binding constant Ki descriptors are molecular properties. The function can be linear regression, random forest, or neural networks. The historical Hanch-Fujita equation from 1962 expressed biological activity as a function of the partition coefficient for lipophilicity, the Hammett constant for electronic effects, and the steric parameter. This pioneering work laid the foundation for computational drug design. The QSAR workflow has several key steps. First, prepare your compound library with SMILES and activity data. Second, calculate molecular descriptors. Third, split data into training and test sets. Fourth, train your model using random forest, SVM, or neural networks. Fifth, evaluate performance. Iterate with hyperparameter tuning until performance is acceptable. QSAR models come in two types. Classification models predict active versus inactive, typically using an activity threshold for the active class. Key metrics include RAW-KUC, precision, recall, and F1 score. Regression models predict continuous activity values. The main metrics include R-squared plus error metrics. Activity values are typically log-transformed. Let's compare machine learning methods for QSAR. Linear regression is fast with high interpretability, but only moderate accuracy. Random forest achieves good accuracy with decent speed and works well for medium-sized datasets. SVM is good for small to medium high-dimensional data. Neural networks achieve high accuracy, but need large samples. LightGBM and XGBoost are competition favorites with excellent speed. Graph neural networks achieve state-of-the-art results on molecular graphs. Real-world performance on CH-embyl kinase inhibition prediction with large training and test datasets shows a clear progression. Linear regression achieves moderate performance. Random forest reaches better results. SVM and neural networks achieve higher performance. LightGBM and graph neural networks using MPNN architecture achieve the best results. This shows the progression of methods over time. An important concept is the QSAR applicability domain. The problem is that predictions for compounds very different from training data are unreliable. The AD is defined as the chemical space covered by training data. Determination methods include Tanimoto distance to nearest neighbor, leverage calculations, and standardized distance. Compounds outside AD need experimental validation. Now let's cover ADMET prediction in detail. Remember, a significant portion of clinical trial failures are due to ADMET issues. In Phase 1, toxicity issues cause failures. In Phase 2, poor pharmacokinetics leads to failures. Predicting ADMET early is estimated to cut development costs by $100 to $500 million. Early ADMET prediction during lead discovery can eliminate problematic compounds before expensive development. For absorption prediction, the key metric is CACO2 permeability, modeling intestinal absorption. High permeability values indicate good absorption. Low permeability values indicate poor absorption. Machine learning achieves high accuracy for CACO2 prediction. Oral bioavailability targets high values. For distribution prediction, plasma protein binding measures how much drug binds to albumin and other proteins. High binding means low free drug. Blood-brain barrier permeability uses log-BB, the brain-to-blood ratio. Positive values mean the drug concentrates in the brain, favorable for CNS drugs. Very negative values mean it doesn't penetrate the brain, avoiding CNS side effects. Predictive factors include molecular weight and polar surface area. For metabolism prediction, CYP450 enzymes are the major metabolic enzymes in the liver. CYP3, a 4 handles roughly half of all drug metabolism, CYP2D6 about a fifth, CYP2C9 about 15%, and CYP2C19 about a tenth, with the rest spread across other enzymes. Inhibiting these enzymes causes drug-drug interactions. Prediction achieves excellent accuracy. Metabolic stability, half-life in liver microsomes varies. with longer values indicating stability and shorter values indicating fast metabolism. For excretion prediction, renal clearance measures kidney drug removal rate. High clearance means rapid excretion. The half-life determines dosing frequency. Very short half-life needs multiple daily doses, moderate allows twice daily, and longer half-life enables once daily dosing. Half-life prediction is challenging because it depends on multiple pharmacokinetic parameters. For toxicity prediction, hERG inhibition is crucial for cardiotoxicity. hERG is the cardiac potassium channel. Inhibition causes QT prolongation and potentially fatal arrhythmia. Very low IC50 values are high risk, requiring development termination. Moderate values are medium risk, needing careful evaluation. Higher values are low risk and safe. This achieves the highest prediction accuracy in ADMET, with deep learning reaching excellent performance. Other toxicity predictions include hepatotoxicity or drug-induced liver injury. Mechanisms include reactive metabolite formation, mitochondrial toxicity, and bile acid transport inhibition. Prediction achieves good accuracy. Mutagenicity tested by the Ames test achieves high accuracy. Structural alerts for mutagenicity include nitro groups, azo groups, epoxides, and alkylating agents. Now let's explore molecular generative models. Traditional virtual screening selects from existing compound libraries, but cannot discover compounds not in the library. Generative models directly generate novel molecules, freely exploring the vast chemical space, and can design molecules with desired properties. Main types are VAE, GAN, transformer, and reinforcement learning. VAE, or variational autoencoder, works with this architecture. SMILES goes through an encoder to a latent space vector, then through a decoder back to reconstructed SMILES. The encoder compresses molecules to relatively low-dimensional vectors. The latent space is a continuous chemical space where you can sample and interpolate. Training uses reconstruction loss plus KL divergence for regularization. The VAE molecule generation workflow is: first train on known molecules, then sample in latent space from a normal distribution, generate novel SMILES with the decoder, check validity with RDKit, predict properties using QSAR and ADMET models, and select favorable molecules. ChemV achieved good valid SMILES generation. Advantages include continuous optimization. Disadvantages include some invalid SMILES generation. GAN, or Generative Adversarial Network, uses two competing networks. The generator creates molecular SMILES from random noise. The discriminator classifies molecules as real from training data or fake from the generator. Adversarial learning means the generator tries to fool the discriminator while the discriminator improves at detecting fakes. Eventually, the generator produces realistic molecules. In silico medicines, chemistry 42 uses GAN-based generative AI. It takes target protein structure plus desired properties as input, uses conditional GAN to generate molecular candidates, then scores them for binding affinity, ADMET, synthesizability, and patent avoidance. This system discovered IPF treatment candidates in just 18 months to clinical trial. The key innovation is WGANGP, Wasserstein GAN with gradient penalty, for stable training. Transformer models treat SMILES like natural language, using attention mechanisms to capture context and large-scale pre-training on millions of molecules. The architecture tokenizes SMILES strings, processes through transformer encoder-decoder, and predicts the next token. ChemBerta, pre-trained on 10 million SMILES, is an encoder. You fine-tune it for property prediction, and it reaches good accuracy from as few as 100 labeled samples. Generation is a different family autoregressive transformers, such as MAL-GPT, and those reach roughly 70 to 90% chemically valid output. Reinforcement learning for molecule generation uses this setup. The agent is the generative model, the environment is chemical space. Actions are selecting next tokens for SMILES generation. State is the current SMILES prefix, and reward is properties of the generated molecule like QED, log-P, and ADMET scores. The reward function combines multiple objectives with weights for multi-objective optimization. Graph generative models directly generate molecular graphs. The process starts from an empty graph, then iteratively adds atoms choosing atom types, adds bonds between atoms, and repeats until the desired size. Methods include GraphRNN for sequential generation, junction tree VAE, achieving very high valid molecules by generating scaffolds, and MoFlow using normalizing flows. Advantages include generating only valid molecules with chemical constraints. Let's cover major databases and tools. CHEMBL from the European Bioinformatics Institute contains millions of compounds and millions of bioactivity data points covering thousands of targets. Data includes compound information, assay types, activity values like IC50 and Chi, and target proteins. The Python API allows easy access for target searches and activity data retrieval. Other key databases include PubChem from NIH with over 100 million compounds, providing free access via REST API with two-dimensional and three-dimensional structures and literature links. DrugBank contains thousands of approved drugs with detailed ADMET and drug interaction data. BindingDB has millions of protein ligand binding data points. Each database serves different purposes in the drug discovery workflow. RDKit is the open-source standard for cheminformatics. Main functions include molecular I/O for SMILES, MOL, SDF formats, descriptor calculation for over 200 properties, molecular fingerprints including ECFP, MACCS, and RDKit-FP, substructure search with SMARTS, two-dimensional drawing for visualization, and three-dimensional structure generation. It supports Python, C++, and Java. The overall drug discovery MI workflow connects all these pieces. Start with target identification, then build a compound library from CHEMBL and PubChem. Perform virtual screening with QSAR and docking to identify hit compounds. Run ADMET prediction for in silico evaluation. Compounds passing ADMET go to in vitro validation. Those showing activity become lead compounds for optimization and eventually preclinical studies. Time and cost savings at each stage are impressive. Target identification drops from 1 to 2 years to about 6 months to 1 year. Lead discovery drops from 2 to 3 years to just 6 months to 1 year, a substantial reduction. Lead optimization drops from 2 to 3 years to 1 to 1.5 years. Overall, the total timeline compresses dramatically. Cost savings include major reductions in virtual screening and experimental numbers. Let's review what you should now understand. For basic understanding, explain 4 types of molecular representations, understand QSAR principles and workflow, explain ADMET 5 items specifically, understand differences among 4 types of generative models, and know features of major databases. For practical skills, calculate Lipinski's rule of 5, calculate Tanimoto similarity, interpret model performance metrics, and select appropriate ML methods. This concludes Chapter 2. You've learned about molecular representations from SMILES to graphs, QSAR modeling workflows and machine learning methods, detailed ADMET prediction for all 5 properties, molecular generative models including VAE, GAN, and transformers, and key databases and tools like CHEMBL and RDKit. In Chapter 3, we'll implement these methods in Python with 30 hands-on code examples using RDKit and CHEMBL.